Hi everyone. It's an honor to be again here after COVID. Right, I wouldn't be able to come here in the two past conference, but it's a pleasure to be here again. So this I have to tell you that this is an ongoing work that we are trying to do back on our lab. We are trying to now bring other disciplines into the code biology theory and try to see how they or can mix and be fixed each other. So what we are trying to do here is to try to see if we can, by the use of network science, by the use of theoretical informational theory and all that stuff, we can try to see how codes are evolving, are going on. So in this case, we are going to talk about a little bit how complexity, information, and entropy can help, how some structures or the life can be go up for those three variables, features, traits, I don't know how you want to call it. So what's going on? First, I remember that when I started reading about the definition of life, one definition that blows my mind was the autopoiesis concept. Because something that it was special about it is that we have this system, open closed system, or kind of not an isolated system, but a closed one that could repair itself. Yeah, but something that it was mixed when I started reading there is, okay, you know that you can build your own cells, but how do you define what are the pieces and how they could be arranged? I remember then reading this work for Vanderlei dos Santos about talking about the concept of semiopoiesis, when they start inferring that there must be something in the molecules that could help you to arrange them. This must be something, it's what we tend to call information. Yeah? Then we have the concept of what we call code poiesis. Yeah? Somehow, all the structures know how to match each other, build and then gather other functions by arranging a structure and then mapping each other by different means. So in this case, what we have is that we know that we have the autopoiesis, then semiopoiesis, then code poiesis, but something that must be gathering all together is that we have information. So what is information indeed? There are some discussion about that information is observable, it's not, it's an artifact, it's only a metaphor, but somehow we know that when we try to define life, information is a trait that always appears. So, okay, what we assume, based on assumptions that the biology then we began, we know that the information is somehow stick in the genome. Yeah? In the genome, there must be the whole instruction bunch of information that give us what life it is. So in this case, if we know that the genome is a sequence, we know that we can have some other traits that we can analyze. For example, complexity. Yeah, we know that the complexity is the measure of how the elements are arranged and how they could be regular, monotony, or can be random. So somehow we know that the relationship between the order and disorder is that when we have an order sequence, we are having a less complex one. But if we go to the other side, we have a very disorder sequence, we also have low complexity. So the complexity is going bigger and then going down. Yeah. In this case, there must be about how once again, we arrange this and what are the microstates or states that we have, and that's the other concept I want to bring on. For example, here we have a very monotonic and regular sequence, low complex. We know we have once a gene sequence that kind to be an orderness, but it's complex. And then we have a random sequence that we know that it's there are no trend, there are only appearing by the same uniform distribution. So this is something disorder. So how can we relate it to a more Boltzmann approach of entropy, meaning that how can we know what are the probability that certain sequences, atoms, or structures can appear. Yeah. So we know that all these three concepts, once again, information, complexity, and entropy, seems to get stuck together when we are defining life. So that must be because they somehow are defining life. Yeah. So what we're trying to see here is if we build this like a space when the axes are those three features, how can we start analyzing and see how the objects, the system, the organisms appears? Because we know that, for example, a bacteria has some information, has its complexity and its level and entropy. But for example, if we look for an eukaryotic cells, maybe we can say that it's more complex, it has more information, or it has more entropy. And also, if we stick together, also in another level, these neurons, for example, we have the brain that is more complex than the other ones. Yeah. So there must be like a path or a way to go from one structure, biological structure, that could be simpler, but has the same functions that someone that could be also complex and fulfill some biological functions. So what we were thinking is that maybe, and we kind of still Rovelli idea of bubbles of entropy, that we have this bubble when you can only have a certain amount of entropy, of complexity, and once you reach that point, you have to escape to a bigger bubble. So in this case, what we are seeing is that we are moving from one bubble to another bubble. What code biology tends to call this bubble is the major shifts of the biology. So we are saying that when we're moving from one bubble to another bubble, maybe we are reaching genetic code. In the next one, we are reaching signal code. And in another level, neural code, and go on until our cultural ones. So maybe evolution somehow tends to choose what are the series of bubbles that we are instead evolving. Yeah? And this based, once again, in entropy, information, and complexity. Yeah? So what I'm trying to say is we already know that we have genome. Genome expresses itself, well, have the mechanisms to express genes, and we have the transcripts. Somehow these transcripts relate each other by building some modular of functions, and this gives us the whole function, a bunch of different functions, and then we have different type of cells, organism, all the stuff. So it's something that it's important here is that we not only have to look for the genome, we also have to look for how interact each other, give us the hint that network science is something important for biology. The interaction of the elements are something that we must take care of and look for. Okay? So, once again, genome, functions, the relationship between these are what we call information. So we have the genome, we found the genes, or that is the approach we tend to follow, and then we have the proteins, and the proteins give us the function. However, what we know is that this is the genotype, this is the phenotype, but something that we are still missing is that the surrounding and the context, the environment, give us also information that tend to change the way how life emerge. Okay? In this case, an approach that we will take to take account about the environment is that we have on the surroundings of the gene some element that regulate the gene. Yeah. In this case, what we want to see is not only the gene, but also the regulatory elements and the circuitry behind this. Okay? So one of the applied approaches that we are having with this is that, for example, we know that we have genomes, and these genomes give us a different type of bacteria. Okay? So in this case, we steal the concept of triphonal codes and start looking, okay, if we chop the sequence and try to see if there is some arrangement, how can we, for example, we already know that the gene has this structure of 3 nucleotides. But what happens, for example, with another regulatory regions? They follow the same pattern, they follow their own pattern, maybe 5 nucleotides, maybe 2 denucleotides or something else. We build this method based on or analyzing the encode regulatory regions when we map it to numbers and then apply signal processing techniques. So we found that there are some features or some peaks that are relevant to analyze. In this case, we know that the frequency is a way to measure periodicity, and periodicity will talk us about what are the arrangement of the sequence. For example, in the case of that sequence, if we found patterns of two, we will talk about the nucleotides. If we found patterns of 4, we will talking of tetra and so on, okay? And that's something that we found, we already published, and we discovered that the sequences of regulatory sequence has a high complexity. So how to start looking at that complexity and see how could we relate and start building the structure and pieces that would give us the codes that start building the organisms. So we now move to natural language modeling or processing, and we say, okay, what will happen if we took the pieces and start looking all of them? Yeah, this is an approach called free alignment. Free alignment is a technique when you took the information, you chop it, and then try to see how the distribution of these sequences are related to the difference we are looking for. So we call all the words that we can find in this element lexical. So we have, for example, 3-letter lexical. We can find 7, 11, and so on. Yeah, in such a way that we have actually a cluster of words related to a function. And what we want to find is how this distribution of words is related to the function, or we could relate it to the functions we have. So something interesting that we found when we started analyzing all the bacteria is that one people will tend to think that when you are looking for the combination of the words, all of them will appear. However, somehow the nature only gives us a few percentage of this. What means that? If you are looking for 3 nucleotides, we'll have the 74 probability. If we're looking for tetra, we will find all of them. But when we are moving, for example, in words of 7, 8, 9, what we were found is that we only use around 30%, 20% of the whole vocabulary that is available. So somehow nature tends to prioritize the words that we are using. So taking that account, what we start thinking is, okay, so we can cluster words that we call informational compartments, but also the way we use these words tends to have a probability of using. So we use a technique called topic modeling, that is a word for natural language processing, and we suppose that each genome is a document. Somehow this document is formed or is composed by different topics, and a topic is a collection of words. These words are tend to use in a specific way. So, for example, if we gather this sequence, maybe we can find those 2 topics. And those topics can help us to taxonomy classify, for example, bacteria. 3 families of bacteria, we already published this. We found that we can cluster a specific topic related to a family. So maybe functions are emerging for this arrangement. But the other thing that we want to also see is that when you are looking for the words related to that family and that topic, we tend to use it in a certain way. Yeah, we are having more common words than others. So this tells us that the structure is not only about having words, but also how we use them. Yeah, and this gives us that, once again, information could be there. Yeah? So we call this dimension of this organization as the pragmatic dimension, because we know that we have a structure, we have the syntax of the sequences, we know that we have meaning that is the function of the sequences, but somehow we prioritize the order to give us how we use the vocabulary to give or achieve such functions. Okay, so in this way, what we are talking about is that if we started with an ambiguous, completely ambiguous generic code, somehow tend to prioritize to the code we have already, the actual and the modern code. Yeah, but this could happen not only with the generic code, but with any code surrounding. Yeah. So what we are talking is that the information has also three dimensions: the pragmatic one, the semantic, and the syntax. We always center our discussion about the codes between function and structure. Yeah, this tells us that we have structures, we have a prioritizing of the structures and the functions. Okay, let's keep that idea there, because we are going to the second approach, or the second example we took. When I was finishing my PhD, I began study the brain. Brain is something that always amuses us, and we want to know how brain emerge. The pathway I follow is that, okay, what happens if I take the expression of the neurons? Okay, so what we do is start building these gene regulatory networks, meaning that we want to know how each gene relate to another based on the co-transcription function. Yeah? So we found that there are motifs in the network called modules, and we see that if we take a look of that models and then see the genes involved, we got specific functions. That means that the way how genes cluster together and arrange itself tends to give us different functions. Yeah? But not only that, if we use these functions and genes to try to see how they relate each part of the brain, we know we can build a new network. This new network is what we call the connectome. Yeah, the connectome. So when we are looking for the connectome, and once again trying to see if there are modules there, we indeed found it. And when we found them, we see that these modules are related to a specific area of the brain. So somehow we already know that there are a continuity between how genes interact each other to how an organ emerges. So we can start thinking that there indeed are a continuity between the codes from the bottom level to, not I don't want to see the higher level, but middle one, because we are stuck in the brain. We are not talking about cognitive or cultural ones, but maybe something like this could happen. Okay. So in this case, once again, let's bring about the discussion about genotype and phenotype and deformation. Let's remember that we are saying that not only how a structure and function are related, but also how the context is involved to defining this. Nowadays, we are studying biology in this way: form and function, structure and meaning, syntax and semantics. And we center and put all our science here. We have, for example, biochemistry more focusing on the form, on the structure, and the syntax. And then we can have theoretical biology, always looking for a structure, but more mathematical ones, for example. Molecular biology is stuck there. But structural biology tends to be in the middle because they want to relate how is the structure related to the function. And then we have genetics that is more centered to the function than the structure, and we can go on and go on. So what we want to do is study them and see that there is also a flow of how we study this. Because if we study from function to syntax, we are having a top-down approach. We are getting function and then try to see how they work. But if we are the other go around, we have a bottom-up. We want to know how a structure gives us the function. Yeah. But let's remember that we are still missing another axis of information. Okay? So my proposal, or the proposal that I'm working on, is that if we want to see how information is clustered and how they relate each other, we have to take account the 3 dimensions. And what we call the relationship between syntactic and semantic dimension is what we call codes and mapping. What we call the relationship between a structure and how they prioritize is what we call evolution. And finally, what we call the relationship between how we prioritize this and give us the structure is what we call the observable, and could be the area that maybe physics or mathematics can help us to develop. And that's it. Thanks. This is the lab where I am part of. So these are my colleagues, students, and another guys that visit us, continue to visit us.
Thank you, Omar.
Yeah.
You have any questions? Yes. Please, Moira.
Yeah, Omar, this is not a question, it's a comment. I would not start with the 3 axes, because you say entropy. This is not, entropy, it was with Carnot.
Yeah.
We have thermodynamic, and then we had non-equilibrium thermodynamics. You are referring to the Shannon in.
Yeah, Shannon Boltzmann.
No, these are completely 2 different things. It's famous. They ask why you are calling this entropy. They say because nobody understands anything in entropy. So whenever I will make presentation, nobody can tell me anything. Okay. So this is on record in the sense that entropy has nothing to do with thermodynamic entropy. So this is a weak spot. You could be attacked. Second, also on the complexity, I mean, depending on which type of definition you want to say, complexity is about the fact that you cannot make a model of it. So you cannot make a model, you cannot measure it. So it's already non-consistent with your selling. And also about the information, again, this is another very tricky thing, because the information is the ability of changing the profile of distribution of probability of a possibility. I don't know what I'm saying, that the 3 axes are very, very slippery.
Yeah.
And
what we are trying to do with this approach is try to fix it to a definition.
Yeah, but what I'm saying is that you can get in trouble if you have an audience, old people like me that have been working in complexity, because that is very weak as a way of defining things, because you cannot define any of the 3 axes, let alone measuring. So if you are selling this as that this is how you measure the thing, is a sort of karakiri, no? Because you start defining something that cannot be defined to measuring. So I suggest the presentation was really nice. I really enjoyed it. But the starting, you may get in trouble with audience of old people like me.
Thanks.
Mario, Marcel, please.
Thank you, Omar. This was a fabulous talk. I was just wondering if you could bring coevolution in the picture, because it seems to me from the very basic level of the lexical that, for instance, Paul-Perrine tracts, why are them underrepresented, and the fact that in virus they are over. So this kind of things. And then I think of Bateson also. Information is differences that make differences. So this proning to interaction that somehow accounts for coevolution, if you have thought about integrating this into the picture.
Thanks. I don't know if it's a question, a comment, or no, just a comment. Okay.
Question.
Okay. Well, in this case, we actually the approaches I put here and the experiments I put here is because we are more like a synthetic biologist, so we want to create our consortia of bacteria that give us a function we want to. So in this case, what we want to do is actually try to put some bacteria we build and then measure how coevolution works with each other and see how, for example, could be related to how the genome is evolving and vice versa, how genome give us or feedback in a positive way the gathering of the function.
Because it's very pragmatical, whether they are a microbiome in the gut or they are exposed to a set of virus that is very particular. So I think it can be top down and.
But thanks.
So we go to the last question, maybe, Robert, and after.
So in the first part, you showed that you checked for different sizes of words, trinucleotides, heptameres, and so on. So these were based on concrete enhancer or silencer elements?
Yeah.
And then my question is, have you checked statistically for other of those word lengths? I mean, the way is we find silencers and we have enhancers. It's known from laboratory experiments. Therefore, we know the sequences. But have you checked for any sequence lengths, let's say up to 15, if there are these lengths that are statistically more frequent? Pattern
we stopped finding after 12 letters words, but something that we did in that time, because it was previous of that, is that try to see if the modids we already found related to some regulatory functions are the length that we are proposal for this. So we don't give that.
The point was the fact that if you find a frequency higher than statistically expected, you find a new function. We don't know, but you can do this analysis and say, so the silencer is not this, but hey, there is something that is statistically highly significant and not expected. This is what I'm pointing toward.
Yeah.
What's the kind of difference in frame shifts of sequence?
We didn't analyze. In the time we moved to free alignment, we didn't care about the frame because we chop all the information. So we don't take account how they are ordered, but if they appear only. No.
So the last one, I think.
I just wanted to ask whether, you ended your lecture with referring to structure and meaning, syntax and semantics. And then you mentioned something that we are missing information. Can you please just elaborate on this? What do you mean exactly by missing information? Just this, because I don't understand.
Thank you. I'm actually not saying that when we analyze the way how biology functions, we are only taking account how the structure are and how the function are and how they relate each other. But somehow we found that there are a way that we tend to use some structures over others. So that missing point is what is the information I call that we are missing, and what in this case I call it pragmatics, because somehow tends to be related to the context of how. So that's why I use in this case like as an artifact pragmatics and say, okay, maybe this is what we can call pragmatics in the biology.
Well, hopefully aspect you were elucidated. So right now we have our coffee break. Okay.